So in the last lecture we have seen when we have run the cufdiff command, which is the cuff differential expression of the genes command, then we are getting the file which is named as gene expression. So this file will tell you if you are having multiple samples. For example, if you are having the control sample and the treated samples, then how many genes are showing a significant difference among your samples. So generally the comparison is done in comparison to the control samples. So for example, if there are certain genes which are showing p-value less than 0.05, it means those are the genes which are having significant difference in your treated samples when compared with the control samples. So for example, as I said before, once we run the cufdiff command, we will get this file which is gene expression file, and once we will click it on here, you will get something like that. Like in column A, you will get the test ID, gene ID, then the gene name, then the chromosome locus, and you will also get the log2 fold change. This is very important, the fold change. So this is the fold change. This is showing what is the fold change in a particular gene in comparison to the control samples. Okay, now you need to remember 3 points. Once the value is 0.8, the expression is high in your treated sample. Once the value is less than or equal to 0.8, then the sample is having less expression in your treated samples, and once the p-value is less than 0.05, there is a significant difference. Otherwise there is no significant difference. So that's what you need to understand. And there is a q value which is a corrected p-value. This is more accurate, but sometimes if you are not getting significant difference in your q value, you can consider the p-value as well. Now we will see. For example, here if you will go to this data and you will click this filter, then this icon will come. Now you will go and apply the number filters where you can see, let's say, less than, more than, greater than 0.8 values. Then you will get only those values which are having higher value than 0.8. So you can see here, so these are the genes which are having higher expression in your treated sample. If you will click on this filter one more time, now every value will come. Then you can apply one more time the filter, and then again you can apply for the number filter where you can say less than or equal to 0.8, and in that case you will get only those genes which are having less expression than your treated samples. And if you will click one more time on this filter, every value will come and your filter will disappear. So before your filter disappear, you can just copy and paste in the another tab of this Excel file and see how many genes are upregulated. Click on the downregulated. You will find the genes which are having which are downregulated in your treated samples. Similarly, if you will apply the filter for the, let's say for the p-value, if you will apply the filter, then in that case you will get the values 0.05. Then you will get only these genes which are having significant difference when in comparison to the control samples. So we can just copy and paste it in the another file. Okay, and then we can just apply the filter so that we are not losing our original file. And this file you can treat it like rename as significant 2. Okay, so now you can see the gene name, you can see the locus, but you don't know anything right now. You don't know what is the function of this gene, what is the name of this gene, you don't know anything. And for doing that kind of analysis, what you have to do, you just go to the internet and click on David annotation. So this is just the basic part I am telling right now. You can go for any other software as well for doing this annotation. Okay, click on Start Analysis. And these are the genes which you can see here are having significant difference, whether they are up regulated or down regulated, that is a different scenario. Then go again on your David annotation, paste it here, go to the upload, paste it here. Then this is the NM file, so I believe this is gene bank accession number. Then click on the gene list and you can just submit. Here you can see multiple accession that you need to find it out which accession actually you want. There are ensemble transcript as well, but I'm going for the gene bank accession because it is starting from the NM. Then you click on the submit list. Once you click on the submit list, you can go to the gene functional annotation tool, click on functional annotation table. Here you can download this file. Save this file on the, right now I am saving it on the desktop. You can save it anywhere on your computer, wherever you want to. So now you will get these files. Okay, now you can just copy and maybe here we can paste. Okay, so but in here you can see the gene ID. This is the gene ID is 175785, and here if you will see the gene ID, it is not 175785, 175785 you can see in column 3 and the row 5. So we need to just look it up. And how we can do that? Before that, just understand what are these things. This is the BP. BP is the biological process. In which biological process this particular gene, because right now you can see the gene name. Gene name is ATPase secretory. Now this gene is responsible for which biological process? You can see the calcium ion transport. Similarly, what is the cellular component? It is present on the Golgi apparatus or on the nucleus. And what is the molecular function? For example, you can see it is the molecular function is the signal transducer, or it is the ATP binding protein, or the structural. You can see the structural molecule activity, or this is, and the fourth one is the protein tag. Now again you can see the CAC. CAC is the pathway. This CAC is a pathway. So in which pathway it is responsible for? Which pathway it is taking part? So you can see it is taking part in the cation transporting P-type ATPase. Similarly for the cell adhesion molecules, for the RNA transport. So this is the pathway in which this gene is going to play a significant role. But now as you have seen here, the ID values in column S and the ID values in column C are not matching. So what we have to do, we need to apply the VLOOKUP. Click on this one, comma, click on this one from here till, let's say here, we will just go till here. Okay, and once you do that, then we want to first see what are the, we want to first match the gene ID. So we will just click 1-0 and then we'll just complete this formula and it will come like this. Now you can see it is 1033609 and 1033609. So right now it is matching. The similar formula you need to apply for the other things also. For example, this is for the gene ID, but right now we are also interested in the biological process. We are also interested in the cellular component. We are also interested in the molecular function. And then we are also interested in a, let's say, KEGG pathway. So then the same formula we need to apply. So we will just apply the dollar sign here. Okay. So we will see copy, and we'll just paste it here. And now we will apply instead of 1, because you can see this is the column 1, this is column 2, this is column 3. So now this is column 4. So we will just type instead of 1 column 4, and it will automatically sort it out. And the same thing we have to do for the CC. So for CC, as you can see, CC is in which column? Column 1, column 2, column 3, column 4, column 5. So we will apply here column 5. So it will automatically do the sorting. Similarly, we have to do the same thing for the KEGG pathway. So KEGG pathway is in which column? Column 1, column 2, 3, 4, 5, 6, 7. So 7. So we will apply here. That is not coming. Okay, so this is not even pasted it here. So we'll just first delete this part. Okay, so we'll just copy this part here, okay, and we'll just paste it. And now, which is the MF? MF is what point? Column 1, 2, 3, 4, 5, 6. So we have to apply here 6. So it will automatically do here. Similarly, now we have to apply for the column for the KEGG pathway. So for KEGG pathway, you can see column 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. So we have to apply here 8. And it will automatically sort it out your KEGG pathway. We don't need it, so we'll just delete this portion. And I think we don't need this as well, so we'll delete this portion as well. And now we will just click, instead of 8, we'll click 7. One, 2, 3, 4, 5, 6, 7. Yes. Now here you can see 2 of the genes are not even being reported in which pathway they are going to take part. So here you are getting nothing. So that's how you can sort it out. So what did we learn today? For example, these are the genes which are having significant difference among your treated or your control samples, and you can see the fold change whether they are higher regulated or less expressed in comparison to your control sample. But at the same time, you don't know what are the genes. This is just the gene ID. In that case, what you can do, when you will go to the David software, as I go here, you can go to the upload, just upload the list. Okay, then you can click on the entries, okay, and gene list, and then you can submit it. Maybe 1 second, you can just paste it. Gene bank accession number, gene submit. So you will get these things. Now whatever the categories you want, for example, I don't want these things, so I will not click on this. Similarly, I'm not interested in any protein domain, so I will just deselect these things. And yeah, but I am interested in the pathways, so pathways are, I have clicked on the CAC pathway. You can also click on the reactome pathway. And the gene ontology, I am interested in the BP, which is the biological process, cellular component, and the molecular function. So you can directly click on these things. And once you are done with this, just click on the functional annotation table, download it, save it on your computer, and type it here, as I have typed it here. Okay. And here you can see, because these columns are not matching, if you see the column number T and column number C are not matching, so we have to use the command which is the VLOOKUP command, as I told you already. So you can repeat this video and see how we have done that. So now we know that this particular gene, for example NM0010333609, is a gene and its name is where we have seen here, yeah. So name I have not clicked on, so you need to just find it out what is this name. For example, you can just see if this is the pathway. So you can just click on the gene name. Okay, you can copy it, paste it. Okay, and because your gene name is at number 2 position, just click on 2 and it will automatically come. So right now your particular this gene NM_0010333609, the name of this gene is chaperone containing TCP1 protein, and its biological process, as you can see here, protein folding. Its cellular component is microtubule organizing center. What is the molecular function? You can see it is an ATP binding protein, and the CAC pathway is not defined, and the name is also there. So that's how you can do the analysis of your gene. And with this note, this lecture is going to be completed. In my next lecture, I will tell you how to make the heat map. Once you are having the higher expression or the lower expression of the genes, then after that you need to make a heat map for your publications or for your presentations. So in my next lecture, I will tell you how to make the heat map. So thank you very much. If you like the lecture, please subscribe my channel, and if you think there is a need of the improvement, then please write in the comment section. Thank you so much for watching.